Morning, good afternoon, good evening. Welcome to the second seminar of our international series of plant breeding seminars. We are honoring women in plant breeding through this series, and it's a pleasure to have you with us, and a pleasure to have Dr. Soraya Bertioli, who is a professor at the University of Georgia, dedicated to peanut breeding and genetics. Particularly, she is focused on the use of wild species to source traits of disease and pest resistance. And she's going to talk about that, the process of creating amphidiploids that can breach into the cultivated peanuts, then studying the genetics of that resistance and using markers for marker-assisted introgression into cultivated species. Soraya has a Bachelor of Science from the University of Brasilia and a PhD from Nottingham University. Soraya, it's a pleasure to have you with us. I'm going to stop sharing and give you the floor.
Great. Well, good morning. Well, thank you, Carlos, for inviting me. It really is lovely to be here, and I'm very pleased that you are creating this opportunity for breeders to talk about their work, and this time for women to talk about their work. So thank you. As you said, I've been working with peanuts, and what I'm going to show is the work that we've been doing for the last 25 years. Part of it in Brazil and part of it in the United States. So starting with where I come from and my story, I was born in Brazil, in Brasilia, which is a lovely city, new, modern. It is a land of opportunity. I was shielded from lots of things that happen in the world. I had a very happy childhood. And one day my parents decided to take me on a road trip to the region where they came from. Because in Brazil, we had something similar to what happened in the United States, which is the Great Migrations. So we took this road trip to the Northeast, thinking we were going to go and see beautiful beaches. And we did. But when we went more inland, I saw things I had never seen before, which is these houses made of mud, and they had no structure. And people were planting beans and rice, and there was a particularly heavy drought, and they couldn't harvest. And obviously, these people were going, hungry. That is something that is not dramatic, but it is it is a problem, and it sort of planted the seed in my mind when I was a child. People are planting, they're not harvesting. What they're going to do? So anyway, so I went on to. When I went to university, I decided to do biology. I didn't know how I was going to work with food, but that was my goal. And I did an internship in Embrapa, which is a Brazilian corporation for agriculture research, very much similar to the USDA here in the United States. And that took me to England, where I did a PhD at Nottingham University. Where I was working with food systems and biological control of pests. So that was really when I really started working with food production. So when I went back to Brazil, I was hired as a scientist to work at Embrapa. And that's where the start of the love story with peanut in 2000. Because Embrapa has the largest wild peanut collection, the most complete in the world. I'm going to show you where peanut comes from in a minute. Just finishing off the story. In 2013, I came here for a sabbatical at UGA, just for 1 year, and life happened and I never left. So from visiting scholar, I became a senior research scientist at UGA, and I have to say it's a wonderful place to work. And all the story I'm going to show you here was done with my husband, and partner in life and work. So whereas you're honoring women, he at least half of it is honoring him as well. So this is what we all did together. So starting why food is important, what we are working with, we all have a common goal here. I think we're all interested in making food better. A lot of us, most of us, have come across Thomas Malthus' prediction. Now, Malthus was an English priest, and he was interested in the spiritual life, but also in the earthly life. And he was interested in food, and he had this terrible prediction that while the population growth is exponential, the food supply is linear. So whereas we have this population growing here at a moment in time, there would be the catastrophe, the Malthusian catastrophe. But what he didn't count on was people being so developed, as ours, developing agricultural practices, fertilizers, defenses, hybrid seed, better genetics, and plant breeding. So the catastrophe never happened. So why did it never happen? And what I'm going to show is the contribution of peanut to this, to keep this catastrophe at bay. So in the last 60 years or so, the peanut yield in the world on average has doubled. It's not the production, it's the yield per area. So whereas in some areas the yield is still very low, we have very, very high yields in places like the United States. So what I'm going to show is a story on how biodiversity and seed sharing can help us feed the world. So what is peanut? No, peanut is a legume, it's a papilionade. It's from a basal clade. So it's much, it's the same family as vigna, as beans, Vicia faba, etc. And the funny thing is that peanut pods grow on the ground. Many people have never seen it. It has a beautiful yellow flower. And when it's self-pollinated most of the time, and when pollinated, the ovule extends to the soil and it buries itself, and the fruit is formed at the end of the ovule. So we have to harvest the peanut underground. So this is one of the tastiest legumes, and it's very important source of subsistence, of food for subsistence farmers. So in the United States, where we are familiar with the production, is heavily mechanized, heavily industrialized. There is a very, very high productivity, and we're all familiar with the peanut butter, M&Ms. So we use peanut. We are in the best place, one of the best places to produce peanut. It's very healthy. We have very low aflatoxin contamination. So whatever peanut is produced in the United States is great peanuts. In other places, however, peanut is not very highly industrialized, mostly eaten in natura, raw or lightly roasted. In places like Asia and East Africa is mostly a women's crop. So women plant and tends to it, and the family tends to it, and mobilizes whole communities of women, and they harvest it and they use the food to feed their children. So in West Africa, on the other hand, is more cash crop. So it varies in different places in Africa. In Africa, they like the red grain, and it's, the whole system is very different. So peanut really permeates a whole strata, different communities. It's produced in many different ways. So the work we do can really impact, big farmers and small subsistence farmers. And just as a note to say, in Africa, peanut is more important, produces more calories than all the other legumes put together. So where peanut is mostly produced? So it's mostly in China. And then India is the second place. Then Nigeria with 8% of production. And United States, we produce only 6% of peanuts. And in Georgia, where I live now, we produce 55% of the total US production. So what we're going to show now is, I just told you what peanut is, where it is produced, and where does it come from. So the origin of peanut, I'm going to talk about, and its wild species. And I'm going to tell you about the domestication, the diploid versus the tetraploid. And the last part is going to be the use of wild species for peanut improvement. So where does peanut come from? So this is the yellow, orange, is the area of occurrence of all the peanut wild species. So peanut has just under 100 cousins, 100 crop relatives. They can be found in many, many different ecosystems in South America. They are in regions, areas where we don't know where they are there. You look in this canopy, you don't know there are peanuts there, but if you look closely, if you have a trained eye, you see these 4 leaflets, little plants, they are all peanuts, wild peanuts. Because they bury themselves, they don't spread very efficiently. A lot of the seed dispersal happens in riverbanks. Peanuts, wild peanuts, also persist in burned pastures. So this is in the west of Brazil. They burn the pasture, so next year revives, and the seeds remain alive under the soil, under the fire, and it germinates the following year. And in contrast, also peanuts, wild peanuts exist in flooded areas. These are marshlands in Brazil. It's called Pantanal, where it's seasonally flooded, and the pods remain viable under the black tropical peat. So we find wild peanuts in many, many different environments, but we do not find cultivated peanuts growing wild. So cultivated peanuts do need people to plant and to harvest and to propagate. So where does the cultivated peanut come from? From this little area surrounded in red, which is now north of Argentina, south of Bolivia. Okay, so that's what we call the Vavilovian origin of peanut. This area is, the east of the Andes. And this is where 2 wild species occur. In the larger part, we see the area of Arachis ipaensis. And there is one population of another wild species called Arachis ipaensis, duranensis and ipaensis. So the Arachis ipaensis happen mostly in the top part in Bolivia. But we believe that people carried one population of Arachis ipaensis and put it in the area where there are lots of Arachis duranensis populations, and they were being cultivated by people, and they got into contact, and they created peanut. And this is how we think it happened. We have Arachis duranensis in that region. We put in green just so to stress they have different genomes. They're not green, they just have different genomes. So they call it green. So we have Arachis duranensis on the top, and we have ipaensis, another diploid. So probably a bee. There must have been a pollinator somewhere, probably a bee, crossed these 2 plants, probably only once. And the diploid that arose from it is sterile. So naturally, it happened a chromosome duplication. This is a phenomenon that is already known to happen in nature. And once you get the chromosome duplication, fertility is restored. So the plant can be propagated, and what we call today a proto-peanut. So it's a plant that it was tetraploid. Probably the people who were having these small gardens saw the plant had probably a little bit bigger seed, maybe. Yeah, the plant was more, as a bigger plant. We've seen it before. We've recreated it. So we had more vigor, and they selected and domesticated it. So they were selected over the years. And we found what we have today, the peanut. again, it's not red and green, but it's just to emphasize that peanut is a tetraploid that has two different genomes in it. So it mostly behaves like two different diploids. These happen over the past about 10,000 years. Okay, so peanut is a very new, it's a very young species. And of course, with that, you see we have a diploid and we have a tetraploid at the end of the story, and they cannot cross, they are incompatible. So peanut, domesticated of peanut is quite lonely. Peanut today cannot cross with the wild species. So it really created a tremendous bottleneck, meaning that peanut has a very, very small genetic diversity. So a little bit of digression here, because I just love this story. Just hold on to this story that it has low genetic diversity. And just let's go to Peru for a couple of seconds. So peanut arose in south of Bolivia, but with the migration in South America, it is found now in Peru. It found itself in Peru. And the civilization Moche from 2,000 years ago were very fond of peanuts. And they depicted in a lot of their pottery things with the shape of peanut. It's really very interesting. And recent excavations found some tombs, and they found what they call the Lord of Sipan. And the Lord of Sipan was a king, a very important king in the area. Lots of gold, lots of silver, lots of power. And if you look closely at his necklace created, it has it's made of peanut, half of gold, half of silver. And the main explanations for it is probably because Lord of Sipan was the lord of the night and the day. It just shows the importance of peanut in many civilizations in South America. Anyway, let's go back to where we are. I just love this story, and I think peanut is a beautiful plant. So the story of peanut, we know in many details now, again, because of botanical collections. So we owe a lot of what we know to botanists, to the collectors. So 3 of them are still with us. The collection started in the 50s. A lot of them are already gone, but these 3 people are the heroes of the day. And also we know a lot of it because of the genome. We were involved in the original sequencing of the 2 diploid ancestors of cultivated peanut, Arachis duranensis and Ipaensis, and of the cultivated peanut. And that happens in 2016, 2019. And of course, the breeding of peanut changed because of the tools created with the genome sequencing. So let's go to where, we are here to talk about, which is the peanut improvement. And what I'm going to show you is the use of wild species, because most of what I do, or what we do, is considered pre-breeding. So we go all the way from the wild species through several processes to get to cultivated, better, improved varieties. So we know that like many other crops, like all other crops, peanut is plagued by pests and diseases. And because peanut has such a low genetic diversity, it really doesn't have a lot to go for. It doesn't have many alleles we cannot get alleles for resistance in the primary pool. So we really have to look outside the box and look at the wild species. Okay, I don't expect you to look at this table, but here, wherever researchers look, wherever they test pests and diseases against wild species, they always find a wild species that is resistant. So this is a work by Tom Stalker. He put together literature review, and for every single pest, there is at least one wild species that is resistant. So we're very happy to see that. And we decided, is our mission, is our goal of the Wild Peanut Lab in Georgia, AUGA, to unlock these resistances and make them available to the breeding community. And how do we do that? So to bridge this ploidy barrier, we have to do a lot of gymnastics. So what we need to do is we mimic Mother Nature, and we make crosses between two wild species. So we use wild species that have complementary genomes. So I'm going to show you a case of a very important plant that has become very important to us, which was a cross from Arachis batisokoi, Arachis stenosperma. So we made manual crosses, many crosses. We created this hybrid, which is sterile. Then what we do to make it tetraploidize is we treat it with colchicine. We didn't invent it. Colchicine is used for gout, so is a medicine for gout, and we use for plants to stop in meiosis. The two nuclei remain together, so it's very toxic. But occasionally, one plant resists it, and tetraploidizes remained viable and restores fertility. So these plants, they recover and they grow, and eventually they produce eggs. This is a process that may take 2 years. So it's a long process until we find a fertile tetraploid seed, from which we have to multiply, and then we start crossing with peanut. And we call them bridges. So that's how we bridge the ploidic barrier. So this is actually an old slide. We had about 15 allotetraploids here. Now we have created over 32, and we are actually surveying most of the allelic diversity of the Arachis section. So the plants, what they look like, they are really very wild. They're not a very good plant. The peanut is a bushy, beautiful plant with lots of branches. And the allotetraploid is very spindly, is very scraggy. So it's not really adapted. When we grow in other places, they're adapted to that region in South America. They're not adapted to Georgia, for instance. They have very small seed. They don't produce many seed. Whereas the peanut has, the isthmus has, in domestication, has become very small. The isthmus that separate the two seeds of the wild peanut and the allotetraploid are really long, and they're not good for harvesting. Imagine you're harvesting, the two seeds are going to break apart. So it's a plant that is not adapted, but it does carry resistance. So whereas peanut is very susceptible, it doesn't have strong resistances. What we want is to hold on to that resistance and make that plant through back process and marker-assisted selection using genetic markers have beautiful, productive peanut at the end of the pipeline. So for that, we do back crosses and use marker-assisted breeding. So I'm going to show you a case study. Okay, so I'm going to talk with a little bit of detail about the use of Arachis stenosperma to bring resistance to root knot nematode. So part of this work was done by Carolina Bajen, who was our PhD student at IPBGG. Now she's in Clemson University. So root knot nematode is a devastating pest. Once it is in the soil, it never leaves. It's very difficult to control, and the chemicals that I use are really not very nice. Reduces production. It's not very nice. So we want resistance, and there is no resistance in the cultivated, in the primary pool. There is one source of resistance, which is Arachis catenaceae, but we want to bring another resistance to avoid the nematode to break the original resistance. So we found, that was a work we did in Brazil many years ago, that Arachis stenosperma is almost immune to root knot nematode. We see Arachis hypogea producing lots of beautiful females after 32 days. Arachis duranensis has some penetration, has suppressed production, But Arachisthanosperma, it's almost no penetration of the nematode. And once it penetrates, the root creates a hypersensitive response, and there's necrosis around the juveniles. So there's absolutely no multiplication from the nematode. So we knew that Arachisthanosperma was a great species. So we created, as I showed before, we created what we call BAT-STEM, Arachis batizocoi versus Asthenosperma, created a population F2 crossing with Arachis hypogea. Then multiplied, we did bioassays, in vitro bioassays, and then with molecular markers, we created a map. And then we identified the regions, the QTR regions, on the chromosome A2 and A9 that bring very strong resistance. Okay, the resistance is almost 100-fold, and we confirmed this resistance in segregants in vitro. Okay, so that's the first part of the work, which is done pretty much all in the lab and in the greenhouse. So what we did with these plants, we crossed and back-crossed, and what we have now in the field, is a photo from last year. This is part of the PhD of Emil Barnes, as a PhD student in the Institute, in our lab. We have a very highly infested field with nematode in Athapolgos, Georgia. So you can see a lot of differences in resistance here. This was the work from these plants. We crossed and back-crossed, and we did marker selection and field selection for 3 years. And once we tested these segregants, the ones that had the markers, segments of A2, once we tested them in the field, we see that the plants that do not have any segment, they are completely detonated by the nematode. This is Georgia 06G, which is the most planted peanut cultivar in the United States, and highly productive, beautiful plant, but no resistance. And we have an advanced line with Arachisthanosperma, no nematode, no multiplication whatsoever. And all the ones that you see that are green don't have nematodes, and they all have the resistance from Arachisthanosperma. And the seeds in the pods are beautiful. So these plants now have gone to yield trials for cultivar development. So this is going to be a first success case story of wild species from the Wild Peanut Lab from Georgia. So while we're selecting for root knot nematode, we observed that these segregating plants also had other resistances. So this is is the segregating graph for leaf spots. And we go, oh, that's interesting. So let's see what else to segregate for. So this was the work from a master's student, Kate Costello. She's now in Texas. So she did detached leaf assays with the segregating population. She used rust. And she found that there was a strong correlation between the presence of the B2 locus from Arachis batizocoi, the same donor, Arachis batizocoi, and rust resistance. So she did bioassays here in Athens, and we got the best plants and sent them to Haiti, because Haiti, they plant a lot of peanuts, but they have a lot of problem with rust. So we thought, well, let's see what happens when they get there. And so we sent a number of segregating lines, some of them without the B2 segments, some with the B2 segments. And as you can see here, highlighted here, a lot of the ones that have the B2 segments, they have very low rust rating. And one of them is called the 226C, has lovely pods, highly productive, and last year we released as germplasm. So it's available to anyone who would like to use them for breeding. So another part of the story, again, with the same population from BAT-STEM, we find that they have some plants have very strong resistance to leaf spots, and to tomato spot wilt virus, which again is very difficult to control, and really reduces productivity. What we found, we put them in field with no fungicide and no insecticide control. And the segregating plants, they have much better resistance than any cultivated peanut, with wild or without wild resistances. So they're really very strong. And this was a photo taken last Monday. Namrata, she's another PhD student in the lab. She's working with this and she's having amazing results. So all these lines are now in yield trials, either preliminary or intermediate yield trials, and they're all in the breeding pipeline. So just to give you a rough idea, again, I don't expect you to read these, but we have a time for development on the left-hand side, done mostly by Carolina Bajen. Then we had testing done now by new students, Kate Costello, Nam, and Emil Barnes. And from now on, we are really doing the latter part of the breeding, which is the yield trials and agronomic traits and nutritional traits. So very soon we're going to have beautiful cultivars. So what I showed you here was the stories that happen in the United States. But we in the Wild Peanut Lab, we have lots of collaborations around the world. So I'm going to show you 4 stories here, very briefly. So 2 stories, or success stories in Africa and 2 in Brazil. So in Africa, this is the work we are doing with Daniel Fonseca in Senegal. So this example here is from 2 wild species, the progenitors of peanut, Arachis ipaensis and Arachis duranensis. We sent these plants to him when he himself was a PhD student. He was doing his PhD between Senegal and France in CIHAD. And from this one single allotetraploid, in collaboration with the breeder Issa Faye, they created 6 cultivars. And this chart here, this photo is a poster they have in the director table in Senegal. And these are the 10 top cultivars that are being produced, they're producing in Senegal. And they're highlighted here 4 of the cultivars that were produced using the wild species. They are in the top 10 cultivars in Senegal in a short duration of time. So they have higher yield and they're much more resilient to drought. And the yield is much more consistent. So it's an amazing story. And we're still working with Danielle, and we already sent many other allotetraploids, and he's been doing great work incorporating these wild species into their local cultivars. Another story is in Uganda with David Okello. So Dr. Okello is a very known breeder in that region of Africa, not just in Uganda, but there's a whole network of breeders in Uganda. And through Daniel Fonseca, who sent population to Okello, we helped with the marker development, with the selections. A population from Arachis batisopus and duranensis, by crossing with a local cultivar, produced between 13 and 32% more than the local cultivar, and the seeds are larger as well. So, and they also have moderate resistance to leaf spots, just moderate resistance to leaf spots, and to GRD, which is a groundnut rosette virus. So this was very encouraging. David Okello is still working with these segregants. We are collaborating in this project through the USAID. And we have 2 students in the lab, Danielle Essendor and Millicent Avosa. And they are now creating new populations using another 5 different wild species. So these populations are being created. We're mapping the resistances, and it's all being transferred to Africa. So again, we hope that soon we're going to be tackling very difficult diseases that they have in Africa. In Brazil, we have two very nice stories. The first one I'm going to show is with Dr. Inácio Godoy. So he's breeding mostly for São Paulo, which is the biggest producer in Brazil. They have very strong problems, very big problems with leaf spots, very much like in Georgia, but it's hotter and it's more humid. And using Arachis cardinacea, some legacy lines, we helped with the marker-assisted selection produce a cultivar called Sempre Verde, which is called Always Green. And the reason it's called Always Green is because it's always green. And at the end of the season, when the other cultivars, if they're not sprayed, they're brown, they've been burned, they're defoliated. Sempre Verde is always green and produces reasonably well. I mean, it hasn't reached the yield potential that is needed for the Brazilian market, but it has a niche in the organic market. And from those lines, we're still in the process of creating new cultivars with higher yield potential. So this is a great story and is an ongoing collaboration. And the second story in Brazil is with Dr. Thais Suassuna, using Arachis and duranensis together. And she created a new cultivar called BRS 425, which is adapted to a new frontier of the production of peanut, which is in the Central West, and very productive with moderate resistance to leaf spots. And again, with Dr. Thais, we're still collaborating and transferring new materials so she can use more wild species. And I think it's a lovely way to end these success stories, because it's like a full circle. I started in Embrapa as a young scientist, and now we are producing cultivars with Embrapa. So it's very, very rewarding to see that this is happening in my country too. And, there's much more, there's many more stories and many more stories to go. We have good students in the lab, in the Wild Peanut Lab, and a lot of other collaborations. So this is just to show the Wild Peanut Lab at Meadville. We have different ways we grow the plants. We produce them in a single plant. When we are selecting single plants, we have mini rows, we have complete rows. So we have a very complex structure of analysis, of evaluating the plants, since we're working with such different stages of development. And just to show you, this is the pipeline. We work all the way from the creation, the use of wild species, the creation of, we call neotetraploids and new allotetraploids. We create into grass lines, we cross and back cross with peanut, and we create elite intergrass lines that we start using in yield trials. And all of this is using marker-assisted selection. And at the end of the pipeline, the goal of everything we do is to create improved cultivars with wild resistances. And the big vision that we have is that we are towards and work towards creating structured germplasm collections. Everything we're creating, we aim to make public. We want to make it public through the USDA. We have in the United States fantastic system of curation, and a lot of it happens in Griffin, Georgia. Dr. Sham Tallery is the curator of the collections. And the USDA has the mandate to hold and to distribute plants to the scientific community. And USDA can also distribute to other parts of the world. We are at the time that we have a lot of restrictions around the world because of the Convention of Biological Diversity, the Plant Treaty, And peanut is under a lot of duress in terms of sharing. But luckily, from the United States, we are able to share. And that's our aim, to produce and to transfer these resistances as far wide as we can, so we can impact billions of people in the generations to come. So with that, I would like to thank the Wild Peanut Lab and our collaborators and our funders. We have very generous funders from different parts of the world, and we have collaborators again in different parts of the world. And the group that we have in the Wild Peanut Lab, and obviously David Bertioli, my co-PI, we work very well together, and we are very open to collaboration. We are a very tight group, and we are very grateful to be able to work with peanuts in an area to help improve food security. Thank you very much.
Thank you, Soraya. Very nice presentation. I'm always impressed when I listen to you because I do have a little bit of my heart in peanuts. I think I told you I participated in when I was back in the university in Uruguay in a collection trip of wild species in Uruguay. And the thing that always remember is that my boss at the time was an evolutionary biologist, and he hypothesized that there was one species going alongside the Uruguayan River and another one inland. And he said, if we look in here, there should be interspecific hybrids. And lo and behold, we found them. It's like, why is peanuts so relatively easy to hybridize with? I mean, among the wild species is always something that I somehow envy for other crops, isn't it? Yeah. All right. Questions in the chat, please. Do you use embryo rescue when making white crosses? A question from Paul here in the chat.
Yeah, we don't. No, we have never needed to. So we have so much allelic diversity in the species we have tried that we don't. So in the Iraqi section, there are 32 different species, and we are going to spend several lifetimes trying to unlock all that diversity. So we haven't had the need. I'm sure there will be a point when there are some species which they have different ploidy. They're not, the base is not going to be 10. Some of them, the base is 8 or 9. So there may be a need in the future, but now we haven't needed to.
Jan is asking if you think that technologies like transgenics or gene editing can help speed this up, for example, via allele mining and identified resistant genes in the wild germplasm?
Well, that's a recurring question. Thank you for this question. Obviously, we think that technology, the more technology, the better. It's great to have the technology. Peanut is very difficult to transform. It's a tetraploid. So, by the time you have 4 copies of the gene, you spend maybe 8 years trying to fiddle with it. So it's not easy. Some researchers have successfully transformed peanut. But we see that it may be cheaper and more efficient to make crosses. Because we are able to make crosses efficiently, we don't feel the need to use transformation. And of course, we have the other situation, which is public perception. People are very against transgenics. Of course, I'm not. I mean, people are very against it. And the peanut funders, they do not fund work with transgenics. So there was a moratorium, maybe something like 15 years ago. So we're not encouraged to use transgenics. But on the practical side of it, I think what we are doing with crosses is it's going to keep us busy for a long time. And now we have the pipeline. We are doing it quite fast.
Yes. Question here from Matthias. Which kind of populations are you using for QTL mapping? Do you develop multi-parental populations like MAGIC, NAM for fine mapping?
Okay, so we other collaborators are doing using MAGIC populations, and they're very good. They're using MAGIC populations on cultivated peanut. Okay, and that is needed to have enough variability for mapping. We are using biparental populations, and we've been very successfully mapping on F2s, F3s, and BC1 F3s. Okay, so that's been because the wild is so different, and the resistance is so strong with most of the resistances we're using. We are able to map in those populations. And we are not doing fine mapping. At the moment, we haven't been looking at genes, specific genes, and there may be a student interested in doing that in the future. Our main goal is mostly now to try to identify the segments, and it doesn't need to be fine mapped. As long as we have a reasonable segment that doesn't have a lot of linkage drag, as long as we can cross and transfer to peanut and associate that with markers, that at the moment is sufficient for what our aim is.
Okay. Please post your questions in the chat. I don't know if it's a question, but you talk that the diversity within the cultivated domain is somehow restricted because of that bottleneck foundation effect of the two diploids crossing and not intercrossing later. I imagine that the introduction to Africa represented another bottleneck on top of the bottleneck. And it happens, for example, when I was working in cassava, it's surprising that cassava in Africa was introduced from the coastal areas of Bahia, which is very humid, and then distributed throughout Africa, even adapting to semi-arid areas, but the diversity was restricted. Is that the same happens with peanut, I imagine?
Oh, absolutely.
In Africa.
That's a great point. So we had peanut has several bottlenecks. So I just showed one. Another one was it's still in South America when we have the plant that is tetraploid, so it is completely isolated from the diploids. Within those lines of tetraploid, you have a lot of genetic instability. So only the plants, because we found later that the two genomes, they're not completely isolated. There's some cross between the two genomes, and that creates some deleterious mutations, and there's a lot of genetic instability. So only the lines that were sufficiently stable were allowed to move forward. So we have a second bottleneck. We described that in a paper in 2021. Then, as you say, which is great, we have a third bottleneck, which is when it was transferred to Africa. Only a very small subset of lines went to Africa. And then we have a fourth bottleneck, it's from Africa to the United States. Because although peanut is spread all over South America and some of Central America, it never crossed through North America. So the peanuts that we have in the United States now, they came via Africa. So there was a fourth bottleneck. So yes, the genetic diversity is extremely restricted. And when we see the diversity in wild peanut, once we transfer it, we can get over 100 times more diversity. So we have the number of SNPs is 100 times higher once we start transferring wild diversity. So it's great about the second bottleneck.
Yeah, I think it would be nice to do some work across species about that intercontinental migration and what it represents in terms of bottlenecks. But Bika, she has a question about you're doing a lot of work in marker-assisted selection, but are you planning to use, or is the program planning to use a genomic selection to increase genetic gain while reducing the time required to develop new cultivars?
Great. Well, thanks for this question. Yes. Well, ideally, yes. So genomic selection, we would be able to do that in this latter phase of the pipeline when we have more advanced lines. And that's only possible then. And I'll tell you why. For the genomic selection, of course, we need large numbers of populations and large numbers of individuals. Peanut has a biological limitation. each plant only produce a few dozen seed. Okay, 100 maybe on a good day. So each plant is restricted in the, maize is great, wheat is great, peanut is very restricted. It starts there. And I'm talking about cultivated peanut in its best performance. When we go back to the wild species, the wild species, there's a reason why not many people want to work with wild species of peanut. It's very hard to grow, it's very hard to produce seed. And although it kind of looks easy, it's not that easy to cross with cultivated. We make it sound easy and we've got a scheme, but it takes time. So this very start, when we have very small number of seeds, we're struggling to keep the plant alive. We don't have enough biological material to do genomic selection. But yes, I think it's a very potentially is be very important in the latter phase of the pipeline.
Sounds good. Okay, well, thank you so much, Soraya. Again, it's been a pleasure and a joy to listen to your talk. Thanks for the attendance. We look forward to seeing you again next Thursday. We have a presentation from Dr. Natalia De Leon from the University of Wisconsin about plant breeding and the infinitesimal model, cause or consequence. We look forward to have you with us, and thank you again. Have a great weekend, and we'll see you next week. Thank you, Soraya, again.
Thank you very much, and thank you everybody for being here.
Yes. Thank you. Bye.